早安，提醒您。Good morning, distinguished guests, ladies and gentlemen, and online viewers worldwide. Welcome to the 2022 Tang Prize Masters Forum in Biopharmaceutical Science. This forum is organized by the Tang Prize Foundation in collaboration with Taipei Medical University. The topic today is using mRNAs as medicine. We are honored to have the 2022 Tang Prize laureates, Dr. Kathleen Carrico and Dr. Drew Weissman with us. They will join us remotely from Philadelphia, the United States. Without further ado, we would like to invite to the stage the president of Taipei Medical University, Ling Huang, to deliver the opening remarks. Ladies and gentlemen, please join us in welcoming President Ling.
Distinguished guests, ladies and gentlemen, good morning.
Good morning.
On behalf of Taipei Medical University, I'm delighted to welcome you all to this forum. It's our honor to co-host this forum with Tang Prize Foundation. This year, 3 winners of Tang Prize in Biopharmaceutical Science are selected. Today, we are very honored to have 2 winners, Dr. Kathleen Carrico and Dr. Drew Weissman, to share their invaluable research experiences with us. Since 2019, the world has been widely influenced by the outbreak of COVID-19. This ongoing pandemic has become one of the deadliest diseases in human history. Therefore, the development of mRNA vaccine is especially important. The new platform development by the 3 winners uses a nucleoside-modified mRNA vaccine. It overcomes 2 challenges people faced, which are innate immune response and the easy degradation of RNA. This ingenious approach breaks through the problems and was the key to the rapid development of the COVID-19 vaccine. It not only saves millions of lives, but also opens a new era of RNA-based therapeutics. Dr. Carrico is an expert in RNA and its chemical synthesis for efficient in vitro or in vivo protein expression. In addition, she has also systematically addressed many related issues to the application of RNA in vaccinology. Dr. Weissman has worked on HIV research with Dr. Fauci's team at the NIH. In collaboration with Dr. Carrico, Dr. Weissman started his research of RNA vaccine. They created stealth RNA that does not cause an inflammatory response. In 2006, Dr. Carrico and Dr. Weissman co-founded a biotech company. Their outstanding research work has built a solid foundation for the development of both BNT and Moderna's vaccines for COVID-19, as well as set the best example of a rapid translation of basic research into clinical applications. This model is exactly the one Taipei Medical University is promoting. We have been actively encouraging our faculty members to translate their research results to clinical and industrial applications. Recently, we also engaged in the development of RNA-related research. We are looking forward to their invaluable research experiences to further spark various inspirations and ideas among us all. I believe through today's forum, everyone will gain a deeper understanding of RNA research development and its future applications. Finally, I would like to thank our distinguished guests and all the participants for joining us today. Thank you.
Thank you, President Ling. Please take your seats. Next, we would like to take a group photo. Please remain in your seats, and our photographer will take a photo from the stage. Everyone, please turn to the stage. During this forum, our moderators will be accepting questions through Slido. Please scan the QR code or click the Slido link below the live stream channel. Thank you for your cooperation. To begin with, I would like to invite to the stage the host of this lecture, Dr. Gong Xingjian, Chair Professor of Taipei Medical University and Academician of Academia Sinica, to introduce our first laureates. Ladies and gentlemen, please give a warm welcome to Dr. Gong.
Welcome, everyone. Never in the history of medical science have we witnessed such an extraordinary impact of rapid translation of basic research to clinics. The successful development of SARS-CoV-2 vaccines by Pfizer, BNT, and Moderna with record speed is just stunning. It took fewer than 12 months, and the vaccines have already saved millions of lives worldwide. This rapid medical advance, however, does not come by easily, and is the result of arduous and sometimes underappreciated efforts of the brilliant and the perseverant scientists involved. The Tang Prize Foundation has honored 3 of them, Drs. Kathleen Carrico, Drew Weissman, and Piet Cullis, as recipients of the 2022 Tang Prize in Biopharmaceutical Science. Today, we are delighted that Dr. Carrico and Dr. Weissman are joining us in this Masters Forum held at Taipei Medical University. They are online to attend this meeting. My pleasure to introduce Dr. Carrico as the first speaker. Dr. Kathleen Carrico is Senior Vice President of Biotech SE. She's also professor at University of Szeged, Hungary, and the adjunct professor at University of Pennsylvania, USA. For 4 decades, her research has been focusing on RNA-mediated mechanism with the ultimate goal of developing in vitro transcribed messenger RNA for protein therapy. She and Dr. Weissman together discovered that the nucleoside modifications suppress immunogenicity of RNA. This groundbreaking work unlocked the opportunity for the therapeutic use of messenger RNA. She has won many awards, and indeed too many to mention. In the past 2 years alone, she has received more than 70 awards and honorary degrees, attesting to the worldwide recognition of her contributions. Today, she will talk about history of messenger RNA development of therapy. Let's welcome Dr. Carrico.
Thank you. Thank you very much, Professor Kung, for very nice introduction. You might mention that for 40 years, I did not get any award. Let me share my screen. And talk to you about the history of the mRNA development for therapy. And it all started with, the discovery of messenger RNA. And as you mentioned, in last year, it became an FDA-approved COVID-19 mRNA vaccine, which is the first one which was approved. So it is 60 years passed, and during these 60 years, a couple of important things happened, which I will talk about, but most of the time it was just painstaking incremental improvement happened. So it started with the discovering of the mRNA, and 2 papers published in 1961 in Nature, both of them emphasizing that the intermediate they found, it is a very unstable. I don't have time to talk about even prior to that in the 50s, how the scientists were looking for this messenger RNA, discovering ribosomal RNA, tRNA, and so on. The importance is that with this discovery, we learn from information flow in every cells in our body, and every free living organism is coming from DNA to mRNA and protein. So the DNA is in the nuclei in form of chromosome in the eukaryotes, and the mRNA is getting to the protein factory, which is in the ribosome, and the protein is produced. At the beginning, of course, scientists could not synthesize RNA. They just could isolate and look at different aspects of it. They identified interesting 5' end in the RNA happened. Furuichi in Japan identified that. And in that year, in 1975, they also, other scientists found that both viral RNA as well as cellular RNA has this interesting 5' end. And if you receive the vaccine from Moderna, let's say, that was enzymatically kept, and that was also in 1975 discovered that what kind of enzyme, viral enzyme, could put this strange structure to the 5' end. Other scientists try to see that whether they can translate the RNA. As I mentioned, they couldn't synthesize, so what they did, they isolated, and they used reticulocytes, which is enriched for beta globin. And when they purified it, they, at the beginning in the 60s, they translated in the cell lysate or injected to frog oocyte. And more interestingly, in 1978, 2 papers was published in Nature where the messenger RNA was entrapped in liposome, so in a lipid, and delivered to primary mammalian cells. And they demonstrated that the protein was made by those cells which normally not express beta globin. So this was very important because this was the first time really messenger RNA was, mammalian messenger RNA was used and with liposome and delivered to cells. Liposome was actually introduced in London by Alex Bankham, and we also were in Hungary, where I was studying and working as an undergraduate student. We were learning about this liposome, and ourselves also we did delivery of nucleic acid, not RNA, but we delivered plasmid DNA to mammalian cells. But it was important as an undergraduate student. I started to work with this. But for my graduate studies, I started to work on RNA, which actually my colleagues here, everybody was organic chemists, happened my supervisor actually made cap analogs for the Furuichi and Aaron Shotkin to identify the cap analogs. But we were working in this antiviral compound, which was just discovered in 77, and this was this 2', 5' oligoadenylate. And so I we delivered also this with liposome to the cells and tried to develop an antiviral compound. As you know, we are in the 70s, and even in the 90s, even in 2000, we just realized that we don't really have any good antiviral compound. But while I was doing these RNA work and liposome, scientists in Harvard University identified finally that maybe the best thing to make in vitro transcribed RNA is phage polymerase. In 1984, they identified the sp6 RNA polymerase, and 1985, the following year, it was T7 RNA polymerase, both one from phage, the virus of the bacteria. And the promoter is very short, and the enzyme fidelity is very high, and then providing the DNA template and 4 basic nucleotide triphosphate, they could transcribe the RNA, and using capping enzyme, they put a cap structure on it. So this was the first mRNA made in vitro transcription and coding for a mammalian human beta interferon. What they did, they injected to frog oocyte, those are large cells, and then after incubating these cells, injected cells, they could find in the supernatant that the medium contained antiviral activity. So this is the first time messenger RNA was made in vitro and coded for a functional protein. So what happened in the following years is that different laboratories started to make in vitro transcribed RNA, and they injected in vivo in mice and demonstrating translation in the animals, and used it for therapeutic purposes, or used this as a vaccine for infectious disease, like 2 papers describing influenza vaccines using messenger RNA, and others started to use for cancer vaccines. But you can see that every year there was just 1 paper, like in these days, maybe every day you have a paper on mRNA. But another interesting thing that those teams which were publishing in the early on, you didn't see that follow-up studies, because the problem was with RNA. And that problem was that very small amount of protein was made, very short amount of time. And as we also learned later, that it was also immunogenic. So what happened the following years is that the translation improvement of RNA, so that from the early 60s, 61, even today, we have RNA performance was increased, purification, better cap, better structural element, modification, and so on, and also together with the formulation. So from the beginning, the liposome, and later other cationic lipid deliveries, and finally the LMPs. So all of these is an incremental increase. While this happening, I started to work with messenger RNA in 1989 when I joined University of Pennsylvania, and it was interesting that we did delivered urokinase receptor encoding mRNA to mammalian cells, and this protein, the encoded protein, is highly glycosylated, post-translation modified, and somehow the cell could do all of these things. So the information was inside the cell, and we just have to deliver this mRNA. So it encouraged me, and when I moved on to neurosurgery, started to make different kind of messenger encoding for these different proteins, antioxidant, and so on. And we did improved translation, and we did a lot of studies. 17 years I worked in this little laboratory here shown. And so I could not see any problem that this RNA would be problematic, like inflammatory, as I want to develop for therapeutic purposes for treating, let's say, a stroke patient. And what happened, I met Drew really at the Xerox machine. And so Drew was interested to make a vaccine, and he will talk about that. And so I made the mRNA, transfected to the human dendritic cells. This was also very important that we used human dendritic cells. And we identified that this RNA is very inflammatory. Of course, I thought that the RNA I was making is identical, which is inside the cell, but it seems that for whatever reason was inflammatory. And one thought was that maybe because we are delivering from outside, and maybe even if we would take out from the cells the RNA and then feed our own RNA from the mammalian cells and then put it to the dendritic cells, maybe those are also inflammatory. So to learn that, what we did, really we isolated different fraction of mammalian RNA from the cytoplasm, mitochondrial RNA, tRNA, and so on. And we tested out in human dendritic cells, added with lipofectin to the RNA and to these monocyte-derived DCs, and measured TNF-alpha as an inflammatory marker. And the RNA which I made, this purple here, was very inflammatory, while other RNA was not as inflammatory, and most importantly here, the tRNA was not inflammatory at all. And of course, the tRNA is very different from the mRNA. It is shorter, different cap, different 5' end. There is no cap on it. But most importantly, we knew that there are so many modifications. Actually, 25% of the nucleosides are modified in the tRNA. And we thought that maybe nucleoside modification is the reason why the tRNA is not inflammatory. But how we could do that? There are so many different modifications here, just here showing the uridine and the pyrimidine in our purine. And the enzymes which were introducing this was hardly known, and not that we could order it. Actually, my fellow student in Hungary, Tamás Kis, who discovered in the 90s that how pseudouridination and 2'-methylation is introduced into the ribosomal RNA. But we couldn't apply these enzymes because it required special structures. So what we did to prove that whether indeed modified nucleoside is responsible for lack of immunogenicity, that we ordered a different kind of nucleoside triples, but which were already, the nucleosides were modified, and we made RNA with it. And 5 of them, we ordered 10, 5 of them incorporated. We were very peculiar about that the RNA, this nucleoside modified RNA, had to be present in the human body. So that is not unnatural nucleosides which we used. And we could make these RNA, and then we tested them out in the similar system we use for the naturally isolated RNA. And what we found is that here the RNA, which was generated without modification, was quite inflammatory, measuring against TNF-alpha secreted by isolated dendritic cells. These were not generated, it was isolated from the blood. But we could see that some of them was not inflammatory at all. And when we looked at closer, we could see that whenever the uridine was modified, then those RNA containing modified uridine, those were not inflammatory. So this is how we come to this conclusion. And of course, the next important question was whether we can translate it. And although some of them was not translatable, with our big surprise that pseudouridine containing RNA was 10 times more efficiently translated than the conventional uridine containing RNA. And learning all of these, and of course we were very happy with it. And what we did is that what usually in the United States they are doing, we filed a patent, we established a company in 2006, and we applied for a business grant, and in which we proposed that we will generate pseudouridine containing RNA coding for erythropoietin, which would be used for treating anemia. So that's what we have done. Here we are using pseudouridine containing RNA injected to the mice, and these were transit complex RNA, and when it contained pseudouridine, this erythropoietin-coding RNA, we could see that even 4 days later, we still could detect erythropoietin in the blood of the animals, whereas the half-life is about like 2 hours. And when we had uridine-containing RNA here in the blue, that quite quickly disappeared. We also could see that it was functional, the protein, because it increased the hematocrit. It's responsible for red blood cell production. And when we injected it weekly, we could maintain high level of hematocrit. And you can see that the amount was very, very small, 0.1 microgram RNA was injected. That was complex with transit. And meanwhile, the uridine-containing RNA, we could not increase hematocrit, but most importantly, the pseudouridine-containing RNA did not induce any interferon. So that was where we had in 2012 and published this. And subsequently, I had to leave the university and I joined Biotech in 2013. At that point, we were not in this nice building. We were on the campus and running up and down. And with Uğur Zain and Özlem Türeci, we started to further optimizing the RNA, codon optimization, optimizing the untranslated region of the RNA, as well as did all of this modification, nucleoside modification, purification. Actually, we worked out with Drew at Penn. So when we did all of these improvements, what happened is now that what I showed you, what happened in the 90s, that small amount of protein, short period of time, now we could make so much more protein from these improved RNA, improved delivery methods, and it was extended translation we could find. And this is we so that, you know, RNA is ready for prime time, and we started to use there to generate bispecific antibodies and injecting animals who are implanted with human tumors. We could generate, we could eliminate actually the already established large tumors by weekly injection of this mRNA coding for these bispecific antibodies, which recognize the T cells and the cancer-specific antigens. And subsequently, we also use this mRNA coding for these different 4 cytokines and injected to cell surface growth tumors in animals, and demonstrated that when these cytokines were injected to a cold tumor, that immune cells rush there, because these mRNA translated to the corresponding cytokines, and then the immune cells were trained, and then when they circulated, they could eliminate even established tumors in the lung. So here is the control RNA, and here is when the cytokines mixture RNA was injected. This study is already in human trial together with Sanofi. So subsequently, I don't even have to talk about, because everybody could learn from the television in 2020, that when we learned about the sequence information of the SARS-CoV-2, that how quickly was developed both by Moderna and BioNTech. Pfizer, the vaccine, which in the same year was injected, and later, the following year, was improved. So many people at that time thought that, oh, this is the first time when nucleoside modified RNA is injected in human. But we who 10 years ago established this RNA therapy meetings, we already knew that many other programs was already advancing in the clinic. One of them was, which was the most advanced, it was for VEGF-A mRNA, which was injected to the heart in patient undergoing elective bypass surgeries. And here is showing in the heart like 30 different places injecting to the heart muscles, naked mRNA coding for VEGF-A, which is, when translated, generate a protein which enhances blood vessel formation. This clinical trial started in February 2018 and now finished the phase 2 trial, and we will see that whether Moderna with AstraZeneca will enter to phase 3 trial. Another study where nucleoside-modified RNA was used successfully in human trial was Cas9 coding mRNA, which was treating patient with amyloidosis, transthyretin amyloidosis. This is a serious disease. Following the diagnosis, patient usually die in 5 years. And what Intellia was doing is that they have messenger RNA, nucleoside-modified RNA coding for Cas9. They delivered the guide RNA together in lipid nanoparticles infused to the patient. And what happened is that this guide RNA directed to the translated Cas9 to the area of chromosome where this toxic protein was produced in this patient. And that already, actually, just last week, we heard again that the patients are not making these toxic proteins. So it seems that even the messenger RNA, which was, might be solving the problem for the gene therapy, because this is already permanent. Changes can be introduced this way. And of course, daily you can hear already in the news that more clinical trial is initiated, not just for vaccine, but many therapeutic purposes. And with that, I would just like to say thank you to all of my colleagues, most of the time I spent more than 15 years working together with Drew and colleagues at Biotech, and who all helped me on the journey. And of course, I have to thank to my family, my husband who supported me all those years, and my daughter, who I am very proud of. She's a 2-time Olympic champion, rower. And thank you for your attention.
Thank you, Dr. Carrico, for the wonderful speech. We'll come back with you with more questions in the Q&A sessions. Next up, I'd like to invite to the stage the co-host of this lecture, Dr. Andrew Huang, academician of Academia Sinica, to introduce our second laureate. Ladies and gentlemen, please welcome Dr. Andrew Huang.
Thank you. It's a great honor to have this opportunity to introduce Dr. Drew Weissman. Dr. Weissman actually kind of stayed in Boston area for many, many years. He got his BA and MA degree from Brandeis University, actually at that time working on with Gerald Fussman. And many of you may not know him, but the Cho and Fussman kind of theory on biochemistry is a very important area. Now, in 1987, he got MD and PhD degree from Boston University. So during those time, I was actually in Boston. And of course, I didn't know Drew at that time. I work at MIT with Alex Rich. Initially, I worked on the tRNA, like Kathleen mentioned. At that time, we learned quite a bit about those modified nucleosides, although I didn't know that all those modified nucleosides are so useful nowadays. But in any event, Dr. Weissman and Carrico, they worked together as a team to develop this really brilliant COVID-19 vaccine for human health. And currently, Dr. Weissman is the professor of medicine at the University of Pennsylvania. Of course, he has been there for many years. And I worked on DNA for many years. Almost 70 years ago, Watson and Crick, of course, discovered the bDNA. And so the name of Watson and Crick associated with DNA has been here now for many, many years. Now, I'm sure that later, for us, Carrico and Weissman will be remembered for this messenger RNA technology, either for vaccination or for therapeutics, will stay for us almost forever. So here I have the great pleasure to have Dr. Weissman to give us a talk on nucleoside modified mRNA therapeutics. Let's welcome Dr. Weissman.
Thank you very much for that wonderful introduction, and thank you for the Tang Prize in Biopharmaceuticals for honoring Katie, Peter, and I. So I wanted to bring up what to me is probably the most critical thing of all basic science. I see from the audience, there's a lot of young scientists. This is the most important thing that you need to learn to be a good researcher, and that's to collaborate. Nothing that's been done with mRNA was achieved by a single person. And this is an overall timeline of everything that's been done that has led to the production of the current COVID-19 vaccines. And if you want to get more specific, to make this vaccine, there was a variety of important first-of-its-kind identifications, all of which needed to be put together to make this vaccine. So when Katie and I started working together around 1998 or so, I was still going to HIV meetings, and I would meet people that I knew well, people like Gary Nabel and Tony Fauci and many others. And they would ask me what I'm doing, and I would sit and tell them about the RNA, and they would listen. And at the end, they would say something like, well, that's interesting, but nobody cares about RNA. It'll never be a useful therapeutic. It's failed clinical trials. Why don't you do something useful with your time? And I would try to argue, and I would point out to them that protein therapeutics is the largest growing element of the pharmaceutical industry. But to make a protein therapeutic, you start with a 50,000 liter drum of CHO cells or other cells that make the protein. Then you have to figure out how to purify it away from the cell culture contaminants. So that means that typical protein therapies, monoclonal antibodies, the most rapidly growing, start at $1,000 a dose and typically cost $100,000 a year for treatment. They're incredibly expensive. And when you want to make a new protein, you start over. You make a new cell line, you make a new purification. So it's a very expensive procedure. And what Katie and I would argue is that RNA was a simple reaction. Moderna, Pfizer use 100-liter bioreactors. 6 liters of RNA reaction makes 1 million doses of vaccine. So small size, small quantity. And if you wanted to change the vaccine, you put in a new coding sequence. And that's what Moderna and Pfizer have done with the new booster. They plugged in a new sequence. They didn't have to do clinical trials. They didn't have to do tox testing. It's very simple. The purification is a one-step procedure. So overall, RNA is incredibly safe, very effective, very easy to make, very easy to scale up. And I'll talk about this at the end. In thinking about vaccines, and I've always been interested in vaccines since coming over from the NIH, this is the outline of where these vaccines started and where we are currently. So the first vaccine that was developed was in the end of the 1700s, when Edward Jenner noticed that milkmaids who were working on cows that had lesions that looked like smallpox were not getting infected with smallpox. And his idea was to take the inside of those cow smallpox lesions and inject them into people. And when he did that, the people were protected. Now, what's important to note is that in this time period, they didn't know about bacteria, they didn't know about viruses, they didn't know about infectious organisms. They believed that disease came from air or food or water or spirits or who knows what. But Edward Jenner was smart enough to figure out that the pox lesions looked the same and maybe they would confer protection. So Katie and I worked for 7 or 8 years developing nucleoside-modified mRNA. And once we completed that, we started looking at a variety of therapeutic developments. In this case, we started working on vaccines. And here, this is an influenza vaccine. And it's a comparison. So what you can see, this is the inactivated influenza vaccine that us older people get. This is the titer of antibodies that you see with this vaccine. This is the live virus vaccine that we give children. Again, this is the titer of that vaccine. This is the mRNA LNP vaccine that we immunized once. This is a titer of 1 to 2,500. It's 5 times stronger than live virus infection. Typically, vaccines never give you stronger responses than infection with the organism. So this caught the attention of pharmaceutical companies and biotech companies that the RNA LNP vaccine may be incredibly potent as well as easy to use and develop. But as an immunologist, I wanted to understand where this response was coming from. When vaccine people make a new vaccine, they're happy if they see a 2-fold increase in titer. So a 50-fold increase really surprised us. So we went back and we measured B cells that make antibodies. And there are different types of B cells. So the germinal center B cells rapidly proliferate, affinity mature, and make high affinity antibodies. They go on to produce memory B cells that are around for the next time you see that organism, and long-lived plasma cells that secrete the antibody in the circulation. We did an antigen-specific assay. So what that means is we took hemagglutinin that was similar to what was in the vaccine and labeled it with fluorescein, and then counted the number of B cells of each phenotype that bound fluorescein, that were antigen specific. And again, we saw about a 50-fold increase in the number of antigen specific B cells of each phenotype. We looked at bone marrow over a year later, and again, we saw enormous levels of plasma cells secreting anti-HA antibody, demonstrating the durability and potency of this vaccine. But we still wanted to understand what the mechanism of it is. So all B cell responses require CD4 helper cells. And there's one particular type of helper cell called a T follicular helper cell. The only job of this cell type is to form germinal centers. And in the germinal center, it interacts with germinal center B cells and supplies help in an affinity-dependent manner. So it selects for higher affinity antibodies. And then it drives them to switch class, to form memory cells, and to form long-lived plasma cells. So we went and we measured TFHs. And in this case, we looked in macaques. And we compared our RNA vaccine to an identical envelope protein with a double-stranded RNA adjuvant. This adjuvant is thought to be one of the most potent at inducing TFHs. But as you can see, the RNA LNP vaccine induced enormous TFH responses compared to the best adjuvant out there. So what that told us is that the RNA LNP vaccine worked from two main mechanisms. The first is that it induces T follicular helper cells that drive antibody responses. And the second is that protein is made for 7 to 10 days by the mRNA LNPs. That leads to continuous loading of germinal centers with antigen, which is also important for generating good antibody responses. We made the HA vaccine and we wanted to test how potent was the vaccine. So as you know, every year we make a new vaccine because the hemagglutinin and the neuraminidase mutate. And every so often, we have a pandemic strain that's completely different from what was around the previous year. So here we vaccinated mice with an H1 HA, and then we challenged them with an avian H5 influenza, so completely different strain. No homology between these two viruses. And we had complete protection with the RNA vaccine. So this again excited pharmaceutical companies because it's a lead up to a universal influenza vaccine. We and others are working on many different vaccines. And what you'll notice is most of these diseases are ones where previous vaccines have failed. HIV, herpes simplex, malaria, EBV, influenza. These previous vaccines in clinical trials have failed. We're also expanding this, as well as others, for vaccines for allergies, food allergies like peanuts, and air allergens like dust mites, vaccines for autoimmune diseases, and cancer vaccines. But to me, one of the most exciting is new RNA therapeutics. So instead of encoding an antigen that you want to make a response against, RNA can also encode gene editing proteins like Cas9, or it can encode genetically deficient proteins like CFTR for cystic fibrosis, or a variety of others. One of them that we started to study was generating CAR T cells. So a CAR T cell differs from a normal T cell. A normal T cell has a T cell receptor that recognizes a small peptide in association with our MHC class 1. And if the T cell sees that peptide, it kills that tumor cell. A CAR T cell uses the signaling mechanism, so it steals the cytoplasmic tail of a T cell receptor, but it attaches a piece of antibody, and that antibody recognizes a tumor antigen. The first developed were CD19, which is expressed by a number of B-cell leukemias and lymphomas. CAR T cells have 1-year survival above 60%, which is an enormous increase in efficacy. But our idea was, in order to make a CAR T cell, it's a very complicated procedure. You have to leukapheresis, so you remove a few billion cells from a patient. You infect them with a lentivirus to deliver a CAR gene. You expand them for 10 plus days to produce trillions of cells. You then give those cells back to the patient. That costs $500,000 per dose. It takes a GMP cell culture facility, which means this can only be done in certain centers, mainly in the US and Europe, with a few centers across Southeast Asia and the rest of the world. Our idea was, we wanted to know, could we deliver lipid nanoparticles to particular cell types? And we accomplished that by binding pieces of antibodies to the surface of the LNP that targeted different cells, and in this case, T cells. So now the LNPs are taken up by the T cells. we put an RNA that encodes the CAR molecule. So the T cell takes up the LNP, the RNA gets out, it's translated by the ribosomes, and it makes functional CAR on the surface of the cell. We put a CAR that attacks activated fibroblasts, and we used a model system of cardiac fibrosis. So what this system does is it makes mice hypertensive, and a hypertensive mouse forms cardiac fibrosis, and that impairs heart function. So gray are normal mice, brown are hypertensive fibrotic mice. You can see that the volume of their heart in systole and diastole increases. The heart gets stiff. And importantly, the amount of blood that it can pump out is greatly reduced because the heart is too stiff and doesn't move well. A single treatment of T-cell targeted LNPs that make CAR molecules return the mice to normal, back to baseline non-hypertensive fibrotic endpoints. And when we went back and we stained the heart, the normal mice had minimal amounts of fibrosis in red. The hypertensive mice had extensive fibrosis throughout the cardiac muscle, and a single treatment of CD5 targeted CAR encoding mRNA LNPs returned them to normal. So we were able to cure cardiac fibrosis in a mouse model with a single injection of mRNA LNPs. So instead of $500,000 a dose and a couple weeks of preparation, we have an off-the-shelf RNA LNP, probably for a few $100s, that can do the same thing. And it can now be used in a variety of diseases like HIV and autoimmune disorders. The other thing that we are interested in is gene therapy. So currently, the way you do gene therapy is you remove cells from a patient. Often these are bone marrow stem cells in kids that have immune deficiencies. You grow the culture, the cells in culture. You deliver a gene editing construct, a Cas9 or another gene editing system, and you fix the genetic defect or you replace the defective protein. You then give those cells back to the patient after you've irradiated them or given them chemotherapy to make space. Again, this is an incredibly expensive, one-at-a-time procedure. It's problematic when you're looking at something like sickle cell anemia. 200,000 people a year are born with sickle cell. They're mainly in Africa and India, but they're present around the world. Ex vivo gene therapy is impossible in Africa and India from the lack of the sophisticated labs and the cost of the procedure. Our idea is that we can make RNA LNPs that target bone marrow stem cells. Here's our pilot experiments. So we've targeted the LNPs with the CD117 antibody. This is the c-KIT antigen that's expressed by repopulating stem cells and progenitor cells. We delivered Cre recombinase to LOXP mice and had 90+% of the bone marrow stem cells gene recombined. We followed them for 6 months and they maintained this incredibly high level of gene editing. We did secondary transplants. So we took bone marrow 6 months after a single treatment and transferred them into irradiated mice. And all of the stem cells and all of the lymphocytes, red cells, monocytes, macrophages, granulocytes from the stem cells maintain the gene-edited phenotype. So we're now looking at this to deliver and treat a variety of genetic disorders in a variety of tissues. We can target heart, lung, brain, T cells, CD4 positive cells, bone marrow stem cells, and we're developing new technologies. I just wanted to finish by talking about something that I've worked on and I've cared about really my entire life. So the worldwide distribution of disease, money, and therapeutics, it doesn't match up. So this is the distribution of the world population. And as we know, the greatest amount of population is in Africa and Asia. When you look where the money in the world is, it's mainly confined to the Americas, namely North America and Western Europe, Australia also, and some Asian countries. But where the people are is where the least amount of money is. And if we pick one infectious disease, in this case, malaria, we see that malaria is also in the region that has the most people and the least amount of money. So what this means is that these countries and these regions don't have access to the best therapies and the newest vaccines and the newest drugs. Africa is still 25 to 30% vaccinated. So what we've done is we're working with a variety of other countries, in this case, Thailand and Southeast Asia, where we produced their own COVID-19 vaccine. The vaccine actually works better than the Pfizer-BioNTech vaccine, comparing this titer to these titers. And most importantly, we built a GMP site in Bangkok that can make the vaccine and distribute it to all of Southeast Asia and beyond. We've also built a GMP hub in South Africa that's now producing GMP-grade mRNA LNP therapeutics. My vision is that these GMP sites, combined with researchers who can make RNA vaccines and therapeutics, will allow regions to make therapeutics that they need. So we know that pharmaceutical companies have little interest in malaria vaccines for Africa, and they have little interest in tularemia or dengue for Southeast Asia. But now these countries and regions can make their own vaccines in their own GMP centers and produce whatever they think they need that they can't get from elsewhere. To me, that's the way we address the inequality of vaccines and therapeutics. So I need to thank the people in my lab and all of my collaborators. Thank you very much.
Thank you, Dr. Weissman, for the amazing talk. Now it's time to move on to the panel discussion and Q&A sessions. For audience, please remember to scan the QR code and type your question online via Slido. Ladies and gentlemen, I would like to invite to the stage the host and co-host of this forum, Dr. Gongxing Jian and Dr. Andrew Huang, to moderate over the next two sessions. I also like to invite to the stage our panelist, Dr. Huang Qianyue, Vice President of the Development Center for Biotechnology, and Dr. Huang Liming, Distinguished Professor, Department of Pediatrics at National Taiwan University, and also the 2022 Tang Prize laureates, Dr. Kathleen Carrico and Dr. Drew Weissman, to join us online. Welcome. Dr. Gong, the floor is yours.
Dr. Carrico and Dr. Weissman, that was just fantastic, very insightful, and very inspiring, very inspiring. Thank you so much. So what we will do is to start the panel discussion and the question-answer period. And we will start with the three panelists' commentaries. Then they may ask you, both of you or either of you, questions. Then we're collecting questions from the audience, whether in person or online. And then I will read the questions to you, and hopefully you will respond to us. So I would like to invite Dr. Ling Huang, Dr. Huang, who is an expert, infectious disease expert here. Dr. Huang is a distinguished professor from National Taiwan University. I'm asking him to provide some commentary.
Okay, thank you, Professor Gong, for the introduction. First, I'd like to thank the organizer for inviting me to this important and valuable occasion to be a panelist. We have heard two excellent presentations from the laureate this year. They are both the experts in the RNA medicine. Every brilliant idea needs a lot of efforts to make it clinically applicable. So from the lecture, we can see that there are a lot of people working in this field to make RNA therapy a reality. So Dr. Carrico has given us a very good example of that. COVID-19 pandemic is a tragic event for a lot of people and for the world. But it also provides a unique opportunity for RNA vaccine to be used in humans in very short period of time. Without this pandemic, RNA vaccine would have to go through a lot of regulatory process to be able to be used in humans. And that process would take almost 5 to 10 years instead of in 1 year. So that's a very fortunate thing for us. SARS-CoV-2 has undergone a lot of mutations with new variants appearing every 6 to 12 months. So now we have Wuhan strain, we have Alpha strain, we have Beta strain, we have Delta strain, and now we have Omicron strains. Every new strain is able to evade the immunity induced by previous strains. So that's why the pandemic won't stop until now. Traditional protein vaccine would have the difficulty to keep up with the antigenic change. As you can see from the second presentation, it's very difficult to make a new protein, and you have to go from the beginning to the end every time. So for every protein, it takes at least 1 or 2 years to make a new protein. But for RNA, it's much easier. So only RNA vaccine can cope with this kind of rapid change and produce the new vaccine that is able to prevent the infection with current VOCs, very no concerns. RNA vaccine is very amazing in their performance. The clinical trial data show that RNA vaccine, when people get RNA vaccine, they produce very high amount of neutralizing antibody, much higher than infection per se, than natural infection. And also in the phase 3 clinical trial, both RNA vaccine by BNT and Moderna, they also both give you a very good efficacy data, 94 to 95%. So, so Taiwan government also have put a lot of efforts trying to develop the biotechnology industry in Taiwan. That effort has been going on for, I think, more than 10 years. From the second talk, you can see that they are trying to help the regions in the world to develop their own capacity to produce RNA vaccine. So I believe the Taiwan government should also pay attention to this field and are trying to make that a reality for Taiwan to be able to produce maybe our own RNA vaccine. I think that's a priority for Taiwan government. And I believe that work has been going. The NHRI now is taking the responsibility. But I'm a clinician that following the large-scale use of the RNA vaccines, a lot of reactions have been reported. Some are expected, some are not expected. The most well-known complication or adverse effect is myocarditis. Myocarditis is inflammation of heart which is not a trivial disease. And the myocarditis usually occurs within 1 week after injection. Young people have the highest rate of myocarditis, and boys are more likely to suffer than girls. There are a lot of mystery in this event. So RNA vaccine is such a new product that has been used in humans for less than 2 years. So the physicians, the scientists, and the lay people, they all have a lot of questions about the RNA vaccines and about what happened after the long term. Maybe after you inject, every year you inject 1 RNA vaccine, or maybe you inject 2 or 3 RNA vaccines sequentially, what would be the long-term effect? So with that, I will raise my question to the 2 laureates. I think my questions are mainly to Dr. Weissman, but Dr. Carrico can also comment when appropriate. The first question is, please comment on the acute side effects such as myocarditis and the long-term safety of RNA vaccine. Is there any explanation about the mechanistic reasons behind the myocarditis, which happens so fast after injection? And the second question is, as a clinician, we observe a lot of immunological problem after injection of RNA vaccine. The most obvious example is the reactivation of zoster after injection of RNA vaccine. And also, a lot of people complain that their old disease that has been so good for maybe 2 years or 3 years and suddenly become worse after injection of RNA vaccine. So I also like to hear the comment of the laureate about the interaction between RNA vaccine and the immune system. So that's my comment and question.
Thank you. So for the myocarditis, I think what's important is the denominator. So we hear about cases, but they occur at a frequency of 1 or 2 per 100,000 immunizations. They primarily occur in adolescent boys, but it's a mild disease that doesn't require extensive therapy, anti-inflammatories, and it passes very quickly. What people don't realize is that COVID-19 infection leads to heart damage, myocarditis, at a rate of about 30 times higher in many more patients. So if you're looking at a 1 in 100,000 risk for a mild interaction versus much higher levels of myocarditis, I think the risk is pretty clear that the vaccine is safe. The mechanism is the unknown, and we and many other researchers are now trying to figure out why this vaccine causes inflammation in the heart. Is it an immune reaction similar to what's seen with streptococcus and other diseases? Or is it something to do with the lipid nanoparticles? There could be many components that lead to this. That leads us to the second question, which is the RNA LNP vaccines are incredibly potent. They induce antibodies that are 3 to 5 times higher than infected people. So they induce very potent responses, and they have high levels of adverse events. 80+% of people have a sore arm. 20% of people have fevers, feel lousy for a day or two, but again, they recover. There have been reported immunologic interactions, but the numbers don't really hold up that there's an association. So clinicians will say, yeah, I think I see more zoster outbreaks, but they're not looking at hundreds of thousands of people to see, are these infections associated with the vaccine and are they causative by the vaccine? So those studies are ongoing. My guess is that the immune activation of the vaccine could induce immunologic reactions to other viruses that are present in the body or that we're exposed to. But again, that's something that we have to study and understand.
Thank you, Dr. Weissman. Dr. Carrico, would you like to comment?
I have to say that I learned immunology from Drew, and I usually ask his advice on these issues, so he knows so much more on that. But I might mention, you know, that as Drew also, and everybody knows, that there is a stimulant in it. Of course, you know, it can stimulate certain things which might be ongoing in the bodies, and then it could be beneficial or it could be not so. So that so many people wrote me that, you know, they have warts, viruses in, and it, after vaccination, disappeared. And it is important, and actually with Drew, we had a review in 2004 writing about that after inflammation and activation of the immune system, you always have upregulation of the feedback inhibitors of the inflammation, which is suppressor, so that you have a suppressed immune state. And this, again, can be beneficial. I have reports that how many people who try to get, you know, conceive a child for years, after the second vaccination, they were successful because some kind of immune reaction went on to the body, and then it was, you know, suppressed in that. And others also that, you know, some pain disappeared, whereas others said that, you know, something reappeared, which was not favorable. So both the immune activation could be beneficial, so eliminating a virus, for example, or it could be when immune suppression also can be beneficial and can be, you know, not so. And so maybe most of the patient is complaining about, of course, the bad effect, and you know, they forget to tell you that how many good things also happened during the vaccination. And people ask me that how they can get more of this vaccine, because now they don't have the joint pain, or they don't, you know, disappear certain things. So, and other effects which like the menstruation cycle disruption, if you can read 100 years ago, the first vaccine also, they already reported that. So that other vaccine also causing things, but because it never happened that 1 billion people get something in the same time. And then it seems that, oh, did you get it? I get it also. And so they are more likely that, and of course the social media and others can accelerate all of this discussion. So that's also a factor in it.
Thank you, Dr. Carrico. We shall move on, and I'd like to invite Dr. Michael Huang, Vice President of Development Center for Biotechnology. It is an institute that bridges academia and the biopharmaceutical industry. Michael.
Okay, thank you. First, thank you, Dr. Carrico and Professor Weissman for a wonderful talk, very inspiring. I'm from the Development Center for Biotechnology, which is also called the DCB. It is a non-for-profit organization established in 1984 with a major mission to help to facilitate the growth of Taiwan's biotech industry. So we are more close to the industrial park part of in biotechnology. We are also develop new drugs, okay? We heavily engage in development of small molecule drugs, antibody drugs, ADC, and also cell therapy, etc. With our, most of therapeutic areas in oncology, immune oncology, etc. I'm very glad to tell you that recently we also add a new research arm in nucleic acid drugs in RNA research that we are not only developing RNA drugs, we also develop other technologies such as LNP technology and also manufacturing technology for mRNA and etc. So we are really want to closely follow the trend of the development advancement of the medicine here. Just as Professor Wiseman mentioned that for the advancement of medicine, it required collaboration. So we have lots of collaboration with academia, with other research institutes also. Our main function is to translate, okay, from the basic research science and move toward the commercialization direction. So that's our main mission. So following what you have presented, I have two comments here. The first is about the great inspiration that your research have brought to our life. Okay? Not only the help saving our life through the development of the vaccine, but also the story of your research itself is very inspiring. Okay? When I mention about this, particularly, I'd like to thank Professor Carrico for your passion for science and for your tenacity and for so many hours working in the lab that focus on the mRNA that has brought a great result to our life. Okay, now this is not only the translation, as our chairman Kong just mentioned, never seen in the science. So rapid translation from basic science to drugs. But in my eyes, it is a transformation. Okay, it's inspiring, transform that the young scientists, our young generation, their mind, that if they work on something that's really good for humans' life, they will get success if you persist on it. And your story just inspires us on this. So the inspiration is enormously. And I would like to congratulate both of you for doing so. And for the second comment is on the work you have achieved is not only made a great success in mRNA, but in my eyes also open a new way. Maybe sometime down the road, it may create a new paradigm for therapeutics. As Dr. Wiseman has mentioned today, there are so many possibilities down the road, and so many new drugs has been developed now. Actually, because we are working closely with the industry, and many investors, many people come here to ask our opinions to want to foresee the future of the mRNA drugs, and also the possible new paradigm for therapeutics. So that it's a wonderful thing to know. Okay. So with this, I would like to ask two questions. The first question will be addressed to Professor Wiseman. Of course, Professor Carrico, you are very welcome to answer also. The first question is, could you think of any alternatives that surpass the efficacy of these nucleoside-modified mRNA treatments? Okay. The second question I would like to address to Professor Carrico. Of course, Professor Wiseman, you are very welcome to answer also. The question is, there are 12 vaccines now approved to prevent from COVID. What do you assume are the reasons for the success of the mRNA vaccines? There are at least two companies, right? Moderna and Pfizer BNT. They have made a great success in the market. So what do you, what are the reasons for success of the mRNA vaccines? Thank you.
Okay, Dr. Weissman, would you like to take a shot of the first question?
Sure. So it's a great question. And you basically describe my life as a basic scientist, which is always looking for something better. Making the RNA better, making the LNPs better, making the vaccine, this gene therapy, the protein delivery improved. We've developed targeting, which allows us to do all of these therapies in vivo. I can't predict the future for what's going to be better. But I can say that basic scientists spend their lives trying to improve on the past, whether it's inventing brand new technology and therapeutics or improving what's already out there. That's what we spend our lives doing. My lab collaborates with over 200 labs around the world on a variety of vaccines and therapeutics. So we don't rely on just our thinking. We like to collaborate with anybody and everybody to make better vaccines, better therapeutics, better gene therapies. Thank you.
Thank you, Dr. Weissman. Dr. Carrico, I think the question was about why your vaccines are so successful for much more than protein or inactive virus or other things.
Yes, I actually, Drew presented the main reason and why it is so successful. You can, and everybody knows, you can make it very simple. It is cheap, it is quick to produce, and of course it is so effective. And so we already know that the lipid nanoparticle contains one component, the ionizable lipid. This is the adjuvant. And it is critical because this adjuvant is not inducing interferon. And the nucleoside modified RNA is not inducing interferon. That's also very important because we already learned that if interferon is induced, then we get much lower level or no antibody at all. And so the success is, it seems like an overnight success, but I can tell that how many overnight we work so hard. And I might mention here also for all of the young students there is that Drew and me, we worked really shoulder to shoulder and with our own hands, so that Drew was generating these cells, which was very novel. I never heard about at that time when I met him about the dendritic cells, that he could generate these human dendritic cells. And then, I was making the RNA, and we educated each other and look at the data, and we get very excited. We try to understand what does that mean, what should we do next, and so we work very much, shoulder to shoulder on those years, and of course we did not understand why others are not interested, but we were working hard on the purification steps, not just the improving constantly the RNA, but how to purify. And Drew is a physician who visited the patients, and then coming back from his medical service, comes to there and did the HPLC machine trial, a new purification and whatnot, so that we did. And we were always very enthusiastic. We always come if something not working and a new idea, and maybe we should try that or that. And it is important that we had fun, important for everybody that they have to enjoy what they are doing.
Thank you so much. You're so inspiring. As I understand, today we also have high school students in the audience. That's also pretty inspirational. Okay, so now I'd like to move to the third panelist, Dr. Andrew Huang, who is academician of Academia Sinica, who is a well-known, well-recognized structural biologist, having worked with nucleic acid structure as before. Okay, Dr. Huang.
Thank you. Yeah, following some of these comments, we have many young scientists in the audience, and I think the stories of two of you indeed are very, very inspiring. For two things. One is, you started with a very basic research in understanding the nucleic acid aspects of modifications and also the influences on the stability and so on. But it is a hard and long working path. And now, of course, many people see the fruition of this research success, but it has been working on for many, many years. Now, in terms of a question of whether or not there are things that we can still do, I think there are still many things. I'm a structural biologist, as Dr. Huang mentioned. Now, the messenger RNA, many people may think as just a linear molecules of these codons in there. However, we do not really know whether or not the selection of codons are important or not, because you can, for each amino acid, you can have several choices. Which one is a better one, and so on? That's a question. Does it have a secondary or tertiary structure that might influence the efficacy of this translation? And that's also another question. Now, how about the proteins that you try to create? That if it involves glycosylation, what are you going to do with it? Do you want to remove it or you want to change it, and so on? So those are some of the comments I have. Now, in terms of questions I would like to raise is maybe on a technical side, for the delivery of this COVID-19 vaccines or other vaccines, do you think, right now, I think mostly we do it by injection, but what about transdermal microneedle patch or nasal spray, some kind of nebulizer methods? Would that be a reasonable choice or even a better choice in terms of the delivery? That's the first question. The second question related to, Drew mentioned that, for distribution of those very effective therapies or vaccines to the kind of less developed countries, now, would let's say, the stability of those materials be important? So do you think one can design better RNA product which may be stored or transport under normal temperature, maybe even 4 degrees or room temperature, so that less developed country can afford to have this material for a longer time? Those are two questions I addressed to you. Thank you. Maybe, Drew, you can, we always target you, huh?
Sure. So, the storage, people remember when the vaccines were first released, and they need to be stored at -70. But what they don't realize is that happened because everybody was in a rush to get the vaccine out. So to do stability studies, that takes 6 months, 12 months. And Moderna and Pfizer-BioNTech weren't willing to do that. They've now done that. So the Moderna vaccine is stable for 2 months at 4 degrees. The vaccine that we made in Thailand is stable for 6 months at 4 degrees in a fridge. And that's long enough to distribute it to anywhere in the world. Arcturus has developed a freeze drying of their vaccine, so it's stable at room temperature. And we're also working with other companies to develop other ways of freeze drying. So I think in the future, stability is not going to be an issue with mRNA LNP vaccines. I think that's been solved, and it continues to be solved as we move into the future.
On this one, I would mention something. First of all, we had stability data because minus 80 Celsius, we already had other RNA samples. So we already knew that for years you can keep it. So you have to realize also that whether you are making a vaccine and you can keep it for 4 years, or whether you have to keep it at minus 20 and then 6 months going through the inventory, and 6 months later you have to throw those out, which is expired. So we already had data for the minus 80, and of course had to generate data for the other temperature. Of course, we were interested too. You don't say that we were not interested to do the stability study. It has to be done with that material, which was eventually get there. So that, higher temperature you keep that, but then it is a shorter period of time. And again, another things, what Drew mentioned, lyophilization, definitely you can keep it higher temperature, but lyophilization is very, very expensive. And also there is a limit, because if the RNA is too long, then also you have to keep it in maybe minus 20. So there is also other components, and you can change the chemistry. You can put there 2' methylation, but you just can see recently was published, it is not translatable. So then you will run into another problem that you don't get protein from it, which would make it. And for the microneedles, with Drew, we were when we were at Penn working together, we met people who made biodegradable microneedle. They even, tested out in my hands and see that we thought that it is such a great idea. But I learned working in the biotechnology that the industry that so which seems a very good idea might be not that so, because the people's skin is different, and you are not 100% sure that whether it get in or how much, and the surfaces. So that there are many nuances there which can influence that how much material gets how deep. I mean, somebody has more fat, and then where it ends up this material, or skin is thicker or something. And so, and also inhalation is, I mean, if lipids gets to the lung, it can be quite toxic. So, again, some kind of nose drops would be reasonable, but, people are working on it, and they already had some results.
Thank you. Thanks a lot. Okay, so now we're opening to the general audience, and I need someone to help me to open up this iPad. So bear with us. There will be probably another 30 minutes, 25 to 30 minutes. I know it's pretty late at your time, but bear with us.
There was another issue was about the codon optimization. You asked prior to that. I might mention that sometimes codon optimization eliminates out-of-frame product, which was for the SARS-CoV-2. Actually, was shown last year that one out-of-frame product was actually is a very good immunogen. And if you codon optimize, you don't have that.
Okay, we shall start our questions. Okay, so we just. Andrew, you and I will take turn. Okay, so let me do the first one. Let's see. Sorry.
Maybe I'll start the first.
Why don't you start?
Well, there are many questions from the audience online. This is anonymous. Asked a question for Dr. Carrico. He or she asked, before moving to the US in 1985, you worked in Hungary, a communist country then. How was it different from working in the US? Was it a hard time for you to work in Hungary at the time? That was a question, sorry.
I work in a biological research center and was very well equipped and a very modern building and a lot of great scientists. And I never wanted to leave, actually. I enjoyed my time there, but we didn't have enough money. So I had excellent environment there. And when I came here, was not so excellent, actually. But I managed, and I tried my best, and I worked a couple of places before ending up at the University of Pennsylvania, where I enjoyed the most at Penn that all of these great lectures. Then the best scientists came and gave lectures, and at that time, in the 90s, there were no YouTube that you could watch any scientist talk any topics. So that the only way it was, even when I was coming in '86, I remember we drove hours to listen to Gallo to give a lecture, in New Jersey, and we went there because this was the only way we could learn about what's happening with HIV and other things. So I like in Hungary, and it was also very, very effective. And many things, we did there, even the liposome delivery we did in the end of the 70s.
Thank you. Dr. Carrico, I guess this is related. I guess people are fascinated with your career. Your academic life was bumpy. Even at your retirement in 2013, messenger RNA drew little interest. Did you ever worry it might never see the light during your lifetime?
Yes, so one reason I went to BioNTech, actually, because accidentally I learned about this company existence, and they had clinical trial. And although I could go to Moderna, also had a job offer, I was thinking that by the time they figure out how to make a GMP clinical trial quality of RNA, maybe I would be dead by that time, because, I thought that it takes longer. And so it was, yes, so I actually enjoyed going to BioNTech and the biotechnology industry. In the academia, I was not successful because there is the promotion, which I didn't get. I get the demotion, and it was, more paper, bigger. So things were important that I was not successful in it. And then when I went to the BioNTech, it was important that we have to have a product which is functional, otherwise we can go home, and everybody worked together. So that was also, so it's a nice thing if people are there in the audience thinking about going to biotech. It is also can be very uplifting.
The next question is for Drew. You said you were curious in engineer and math from the childhood, probably in Lexington, right? Later, you went to study immunology. What influenced you to change from machine to people? That was the question.
It's a great question, and I probably don't understand how that decision was made. Part of it was every day of my life, my mother said, you can be whatever you want as long as it's not an engineer. So I think she helped influence me into medicine. I think the reason I chose immunology is because back then, and this is a long time ago, but it still remains now, it's really undiscovered country. There's so much we don't understand about immunology that every morning I wake up and think about what people reported, what new studies show, and wondering, are they right? What are we learning from what they're doing? What are we learning from what we're doing? So it's constantly addressing new problems and trying to develop new ways to understand. And that's what excites me the most about immunology.
Thank you.
Thank you. I think Jing is asking, I think both of you, the infants born during pandemic might have poor immunity since they are raised under quarantine and mask protection over decades. What situation do you think we may face in terms of these newborns?
So I guess I can start. I'm an allergist by training, and there's a field of allergy that believes that allergies form due to lack of exposure. So when people moved out of inner cities into suburbs and stopped being exposed to antigens and other things, allergies increase. So certainly being born during the pandemic, where you're locked in a house with only your parents and maybe a sibling, your exposure is going to be much lower. And I wonder whether those kids are going to have higher levels of allergies. I think as far as infections go, they should be fine because they're exposed from their parents. The quarantines were months, not 10 years. So I think they've been suitably exposed, and I wouldn't have a problem that they're going to be immunosuppressed to infections. But the allergies concern me.
Oh, okay, thank you.
Yeah. Well, the next question is from Ling Hong. It's a long question, so bear with me. Professor Weissman, just like you said, mRNA vaccines can directly stimulate immune cells in Gemini Center and help generate better immune response. My question is, before 10 years old, lymphocytes would continue generating and educate by bone marrow and thymus. This young lymphocytes would tolerate all antigens. I'm not sure whether this is right. They attached before educate after they become circulating lymphocytes. What is possible effect of mRNA vaccines for immunity for, well, low age, I think should be a young age receiver? That was the question. Sorry, it's a long question.
Yeah, so I'm not sure I completely understand, but we've looked at immunizing baby mice, baby hamsters that are a couple days old with RNA vaccines, and they make very potent responses. So after we're born, our immune systems, even though they're naive, they're ready to respond and make effective responses. Typically for humans, the first 6 months of life, you have maternal antibodies that give you extra protection against a variety of diseases. But if you're exposed during that time to a strong vaccine, you'll make a response. If you're infected with a strong pathogen, you'll make a response. And that response will stick with you for much of your life. So our immune systems are certainly naive, but vaccines don't result in induction of tolerance when we're young. We respond to them, we make protective responses. So I'm really less concerned. And we've seen the same in fetuses. When their mothers are immunized, the fetus is born with maternal antibodies that protect them from that disease. They don't become tolerant to the disease.
Okay. It just jumped, sorry. This is related to your question, actually. I'm curious about shelf life and the storage of messenger RNA vaccines, as they are more sensitive than protein. are there possible improvements? I thought you have somewhat respond to that. Okay, so sorry, let's move on to the next one. How do the messenger RNA therapies such as nucleoside modified RNA would suit the concept of precision medicine in cancer research? Is that too vague a question? I guess, how do you direct a personalized therapy?
I mean, Moderna is using nucleoside-modified RNA for cancer vaccines, whereas BioNTech using conventional RNA. Because for cancer vaccine is not the antibody, but more the cellular immune response is important. And so it might be not important. We have to see. Because, you know, the generating a cancer vaccine is much more challenging because for the virus, it was obvious that it has to be targeted spike protein, but for the cancer, it is, there are cancer antigens, but not necessarily those are the drivers, and they, you know, even if it is eliminated and some cells will die, you know, the tumor might still grow. So that to figure out exactly that what should targeting by the cancer vaccine is still, you know, in the progress right now. Constantly we are learning, and so is the vaccines is getting better and better, and but more learning is needed. But considering whether it is nucleoside modified or not modified might be, doesn't matter. We have to see.
Okay, thank you. Andrew.
Okay, the next question is related to the modified nucleoside. In 206 to 208, why don't you use N1-methyl pseudouridine as main modification instead of pseudouridine? And maybe I'll add a little bit further questions. Those modified nucleosides, my understanding is pseudouridine, for example, is produced in large quantity in a Japanese fermentation company or something like that, and from microbial source. So this kind of very specialized microbial source, are they available in other countries? Or, you know, if we want to make mRNA modified nucleoside mRNA, we need to get those materials as well, right? So there are two parts here. One is N1 methylated pseudouridine versus uridine.
As I mentioned, we were using what was commercially available. And we were limited only that we will use what is naturally occurring in the human body. Because we already were aware of the 5-position iodinated, like called phthaluridine, for example, which was not toxic for mice, not toxic for monkeys, but 5 volunteers died out of the 15 when they tested out for hepatitis. So we wanted to use nucleoside-modified RNA, nucleoside-modified triphosphates, you know, so we had to get already triphosphate, and so, as I mentioned, we purchased 10 different ones, and of course, you know, whatever incorporated, that was selected. And even in our 2005 paper, we already showed that the 2' momethyl, the pseudouridine, the 2-thiouridine, and 5-methyluridine, that all of the 4 is reduced immunogenicity, so that we knew that uridine modification was required, and these were the commercial available at that point. So in our patent, we listed all of the nucleoside modified naturally occurring in the body. And so as I showed in my slide, you know, for uridine containing, you can have like, you know, 10 more, but it was, we already could see the pattern that that's the rule.
Thank you. I mean, if you look at our patent, the claims list N1-methyl pseudouridine as a modified nucleoside to use for RNA therapeutics. So we thought about it in 2005, but we had to wait for a company to make the triphosphate version in order to test it.
Thank you.
So the question is, hello, I'm curious about the BNT and the Moderna vaccine, both using messenger RNA skill, but why the protection time was so very different? Is that true or not? I don't know whether it's a company secret or not. I don't know.
No, the durability is a little bit different. And I think you have to remember that durability is dose related. And Moderna gives over 3 times more mRNA LNP vaccine than BioNTech. The amount of difference is small. So, you know, it isn't a big difference, and it might be dose related.
Thank you. Yeah, Andrew.
Okay, thank you.
2 or 3 more questions.
Yeah. The next question is from Jiwei Guang from DCB. This is directed to Dr. Weissman. Do you expect the LNP mRNA can be realized for monoclonal antibody production in large scale with efficacy better than human PK? I'm not sure what that means. But anyway, would it be a safety concern? Can we, in other words, I believe the question is, can we use this technology for monoclonal antibody production in human?
Yeah, so we published that 7 years ago using mRNA to make monoclonals. BioNTech published making bispecific antibodies with mRNA. Moderna has already completed a phase 1 clinical trial delivering chikungunya neutralizing monoclonal antibodies with mRNA. So they're already in clinical trials and being developed.
In BioNTech also actually for cancer-specific monoclonal antibody. This is also delivered as an mRNA. It is in clinical trial.
Right, thank you.
Okay. Yeah, maybe, yeah, 1 or 2 more questions, yeah. Okay, I don't know whether Dr. Weissman really, yeah, okay. The question is direct toward Dr. Weissman. I wonder that what's the difference between the vaccines generated in Thailand versus Western Europe?
So I have to be careful because I work with BioNTech, but as I showed in our data, the Thai vaccine actually gives a better antibody response and a better T-cell response. There's probably a few different reasons for that. We use a different version of the spike, and we use a different LNP. And both of those are critical for amount of response you get. But when we compared head-to-head with the Pfizer-BioNTech vaccine, and the Thai vaccine gives higher antibody titers.
Okay, Andrew, maybe the last question.
Well, there are actually many questions.
Yeah, there are too many, and we really apologize. There's so much enthusiasm on this, and we really hope that you can come to Taiwan next year in person. Truly, it's a, I think Tang Price Foundation would really love to invite you to come.
Do we want to open a couple questions from the live audience?
From the audience?
Yeah.
Okay, maybe 2 questions, yeah. From the audience.
Yeah, we can open 2 questions from the audience. If anyone, students or whoever want to ask a question, we can relay the question for you. If not...
Sorry. Oh, so they also already, yeah, they put it in here. But I think he was asking whether people, in the last 2 questions, whether people want to raise hands and do that. Okay. So I guess the lights, so we can't see the audience in the back. But anyway, maybe one more question? I can't. Anyone raise hands?
It's quite late in the East Coast of the United States.
Maybe you have the final question. Yeah.
Well, I don't know. There's so many questions here.
Yeah, there are too many questions. I'm really sorry that, okay.
Well, this concern about the safety, I think Drew already mentioned it's quite safe, but I think people still worry that after you put this messenger RNA into your body, and one question is they are not specific to an organ or to a place. And so they wander around in the body. Would that create potential damage or side effect on our system? That was the question.
You know that for years we had to answer the question that, oh, it is degrade, it will be gone so quickly. And now that all of a sudden, you know, with the vaccine, oh, it might be stay around, you know, it degrades. But
okay. Well, people actually mentioned, I don't know if it's true or not, somehow the messenger RNA create a DNA form into the product. Is that true?
No.
It's impossible.
Isn't there some reverse transcriptase inside the cell?
Even if there, you know, even if you got into a positive cell that had a reverse transcriptase, there are no sequences that the RT can recognize to make a copy. And you know, it's a normal mRNA. So we would see our genomes full of mRNAs if that was possible. So that it just doesn't happen.
And we don't have a mechanism, anything in our body that the RNA would go back to the nuclei. There is not. You need viral proteins recognizing specific sequence to take it.
So thank you so much. I have to conclude this live streaming. It's also a first experience for us. It's a little bit nervous here. But thank you so much, and thank you for your very, very fantastic lectures. So inspiring, so insightful. And we really hope we can see you sometime in person, perhaps next year. And thank you for staying up late. It's really late. Good night and have sweet dreams. Okay, so
thank you very much.
So we conclude this lecture. Yeah, 我们就到这里结束。谢谢各位，谢谢各位参加。
Thank you for host and our panelists. This concludes this session, and we want to thank all forum speakers, moderators, and participants. It is because of your participation that make this forum a success. So please remember to return the audio device you borrowed, and we also have a lunchbox for you to pick up at registration with your lunch voucher. And thank you all, and we wish you a lovely day. 堂讲第5届大师论坛生技医药厂到此圆满结束，请台上贵宾们与我们的堂讲基金会执行长合影。那接续我们也邀请台北医学大学林建煌校长、陈瑞杰董事长、张文昌董事、何鸿能总顾问、陈振宇副校长、吴麦思副校长以及李刚远执行长上台与我们贵宾们合影。谢谢各位来宾的参与。提醒您离开前，请留意随身物品，如有借用，请将翻译耳机还到报到处旁租借服务区。报到处有简便餐盒供来宾们凭餐券领取。再次谢谢您的参与，祝您有个美好的一天。